Okay, today we are doing the molecular diagnosis of leukemia. This is diagnosing leukemia using so many different methods. That is the doing lab tests with the bone marrow and the blood tests, and doing so many different forms to find out the best way to diagnose leukemia. These are some of the ways to test for the presence of leukemia. That is lab tests, flow cytometry, biopsies, imagery tests, pulmonary malfunction tests, lumbar puncture, and molecular tests. These are some of the examples of how these tests look. This is normally the bone marrow testing. You can see over here they put, they take it from the hip bone. There's a hip bone and the injection goes in. In the bone marrow testing, you can take a liquid, or you can take the small piece of the bone with the liquid present inside. This you can do blood tests, because blood tests can pick up leukemia also, especially CML, chronic myeloid leukemia, can be picked up easily in the blood tests. For other types, you would have to test further. As you can see, this is how your normal blood should look, and this is how your blood looks when you have ALL, that's acute lymphocytic leukemia. This is one of the pulmonary function tests that you can take when you are symptomatic of leukemia. This is where you have to measure your inhalation and your exhalation via this machine. Okay, lab tests. To diagnose a person with leukemia, the first procedure is a simple blood test. From if you do not see anything from the blood test, then you go into further testing, such as bone marrow testing. In the blood test, they can do a CBC, that's a complete blood count. They can count your platelets, your white blood cells, and your red blood cells. And a differential helps to see what types of white blood cells are present in your blood. And the peripheral blood smear is just a drop of your blood that is being spread across a slide, and this is looked under a microscope. And from there, they can see a more detailed configuration of what you may or may not have. Bone marrow tests can be done through biopsies or aspiration. The aspiration is the liquid form of the bone marrow test, and the biopsy is the taking part of the bone with the liquid marrow inside. And when you get this, the bone marrow test, you can see things such as gene changes. You can see things such as chromosomal and cell changes in the body and in the bloodstream. This is how a CBC looks like. Over here you can see they have the red blood cells at the bottom, the white blood cells and platelets over just above the red blood cells, and they have the blood plasma, and they send this thing in for testing. Over here you can see is a differential WBC test. Over here you can see there are so many different forms of white blood cells. As you can see, neutrophil, eosinophil, basophil, monocyte, and a lymphocyte. This is what you would know as a blood smear. As you can see, it's one drop of blood that's spread across a slide. Over here is a bone marrow aspiration and biopsy, and how they are carried out. Flow symmetry. This looks at a closer look at the DNA to tell whether it is a normal set or abnormal set of DNA. In this way, you can find out how fast the tumor grows, what size it is, and what shape it is. they normally do a bone marrow biopsy to look into more detail in with a flow cytometry. Over here, you can see you can find out how big your tumor is and which place it's situated in, and which surgery you can do to help remove the tumor, or whether you would need to use radiography, chemotherapy to remove it. Image testing, this is it allows you to see a more visual image of the inside of your body. Over here you can see your lung. You can see if there's any infections on your lung, if there's any infection around your body. You can see for infections in your brain and your spinal cord. With all these imaging, you have a more visual understanding on what is attacking your body. Over here we have x-rays, CT scans, PET scans, MRI scans, ultrasounds, and echocardiograms. All these different ways allows you to get a better understanding. This is what an x-ray looks like. This is what a CT scan looks like. This is what a PET scan looks like. This is an MRI scan. This is an ultrasound. And this is an echocardiogram. Normally the ultrasound and the echocardiogram works hand in hand, and the rest of these are done with radiography. Then we have the pulmonary function test. Over here you can see there are 3 different types of the pulmonary function test. They have the spirometry, the body plethysmography, and the diffusion capacity. Over here they use a machine to measure the inhalation and the exhalation of your lungs. Over here the spirometry, it shows you need to inhale as much air as you can, and that is being measured, and to check if it's at a normal rate. The body plethysmography, it measures the inhalation rate and your exhalation rate and your lung capacity. The diffusion capacity, you have to exhale as hard as you possibly can, and they will measure and see if it's within the normal standards. Then we have a lumbar puncture. This is an injection this is when the cerebrospinal fluid is taken out of your back. It's sort of like an epidural. It's slightly different than that. It extracts this fluid, and it goes for testing so that you can see the extent in which the leukemia has spread throughout the body. This is also very helpful when diagnosing for leukemia. The molecular testing of leukemia, it normally focuses on karyotyping, fluorescence in situ hybridization, DNA sequencing, and PCR. A few examples of these. Karyotyping is directed towards chromosome number, structure, and other abnormalities of the blood test. This can use to detect whether the leukemia is hereditary, and you can see if you were a carrier, or if you came from a carrier, or if you would have it. FISH, this is detecting how the chromosomes work in your body, and with the DNA sequence and so forth. And this uses fluorescence light so that you are able to see where the cancer cells are highlighted in. You can see the location, and you can see where they are, and how many they are, and how big they can be. DNA sequencing, this is the order of the nucleotides, and with the nucleotides in any different other order can make a whole new protein. The bases are adenine, thymine, guanine, and cytosine. Adenine and thymine are usually together, and guanine and cytosine appear together as well. Okay, this is what karyotyping looks like. This is a representation of the chromosomes in the body. It has 23 pairs of chromosomes, 22 pairs somatic cells, and the X and the Y chromosome. As you can see over here, this is a female. DNA sequencing somewhat looks like this, and over here you can also tell with the abnormal sequencing, you can tell whether a patient could have leukemia from just looking at this DNA sequence compared to a normal patient. Fluorescence in situ hybridization. This is using a fluorescent dye, and over here you have to, it's also one of the steps in PCR. Okay. The PCR is a polymerase chain reaction. This makes use of tiny segments of DNA. It amplifies it to millions and millions and millions of DNA. The amplification of this DNA allows us to understand the science and to allow us to study this more intricately. There are so many different stages of PCR. PCR is used throughout the entire medical industry, and it's being expanded throughout the years. PCR is one of the best reactions to be working with right now in terms of medicine and chemistry. PCR techniques allows millions of DNA to be created from specific segments, and this can be used to detect the presence and absence of genes to identify pathogens. With PCR, you can detect various sicknesses, various abnormalities in the bloodstream. You can detect early symptoms. You can detect cancer earlier than normal. It is a very useful technique. This is also used in forensics as well. The PCR setup requires the duplication of the DNA template, the primer initiation, DNA nucleotide bases, the Taq polymerase enzymes, and the buffers under the right conditions. This is how the PCR works. You have to do a blood test to get the extraction of this DNA to work with that during the PCR. In the PCR, they have so many different methods, and there is a thermal cycling system that works throughout the PCR. The temperature is it increases and it drops rapidly throughout the entire process, and the enzymes obviously do their work when that is happening. It works in sync with each other. The main stages of the PCR is the denaturation, the annealing stage, and the extension stage. The denaturation is when the DNA is separated due to the extreme high temperatures in the body. The annealing is when the DNA primer attaches to these DNA segments that have been separated. The DNA primers then attach themselves to each end of these segments when the temperature is lowered again. And the extension, a new strand of DNA is then created via the Taq polymerase enzyme, and this happens when the temperature is raised again. This procedure happens throughout. It happens for hours. It happens as long as DNA is being made. The copies of DNA are being made, and you only need a tiny segment of DNA to start this initiation. The electrophoresis, this monitors the quantity of the DNA, how many DNA is made, the size, how big and how small these DNA segments are produced. and this process is repeated over 30 to 40 times, and it takes a few hours. In certain cases, the DNA, this PCR reaction can go very quickly with the depending on what types of machines you are using. Obviously, if you're using a better machine, this PCR will go a lot faster than normal. These are the components of PCR. This is the template DNA, the primers, the DNTPs. The DNTPs are the adenine, thymine, guanine, and cytosine. Then the Taq polymerase is the enzyme. The buffer, depending on what condition is present, and the PCR tube. PCR tube is very tiny and it fits in a tray. And over here you can see the three basic steps. The denaturing of the DNA. It separates the annealing, the primers attached to either end of the DNA segments, and the extension when they rejoin. Okay. During the diagnosis of leukemia, PCR is usually the DNA for the PCR is taken from the bone marrow, either the aspiration or the biopsy. The PCR can distinguish between the different types of leukemia. It can distinguish between acute lymphoblastic leukemia. It can distinguish between chronic myeloid leukemia and so forth. The PCR is able to identify which type of chromosomes are present in the blood to detect which type of leukemia can be affecting the whole system. With the PCR, you are able to detect leukemia at an early stage, and you can have treatment options for that specific type. With PCR in diagnosing leukemia, the survival rate is very high. If normally people with leukemia have very few years to live, but with using PCR detection for the leukemia, you can increase the chances of living. You can have a longer lifespan. You can increase the quality of life as well. And you can find the direct treatment plan that will help instantly. PCR works hand in hand with DNA sequencing, with fluorescence in situ hybridization, and with karyotyping. All these work in sync so that you can get a more accurate answer, and you can analyze the genes more clearly, and you can find your answer more faster in diagnosing leukemia. Over here you can see they are using karyotyping from the PCR reaction. The PCR reaction is done here in this diagram, and this information from the PCR is then put on the karyotyping, and from here you can distinguish between a person who's normal and a person who has leukemia. Over here is the negative PCR control. This is a healthy control, and this is acute leukemia patient. As I said, it works all hand in hand. Okay, there are some of the references that I used for my presentation. Thank you.